Welcome to Hartline Biology, reminding you that biology has its own language, and we love to name everything in biology. So, learn the language, folks. And please subscribe to the channel. Also, you can find free flashcard links in the video descriptions. Just a recap of some of the fundamentals of DNA replication. DNA is a double-stranded molecule that separates in order for DNA to replicate. Then new DNA is built to complement to the existing single strands of DNA, and DNA is built from 5' to 3'. This happens on both of those single strands of DNA to produce two identical copies of DNA. Also reminding you that DNA is anti-parallel, where one strand goes from 3' to 5' and the other strand goes from 5' to 3'. There's a bunch of enzymes involved in DNA replication, including DNA helicase, which unzips double-stranded DNA to form single-stranded DNA, and topoisomerase, which uncoils the helix of DNA to prevent supercoiling, enabling DNA helicase to continue separating the DNA. We also have enzymes that help to build DNA, including primase, which actually builds an RNA primer. Then DNA polymerase III will extend that primer to produce new DNA. The RNA primer is then removed by DNA polymerase I and replaced with DNA. Once new DNA is formed, you end up with these little gaps known as nicks. And we have an enzyme called DNA ligase that forms covalent bonds between those DNA fragments to make continuous new strands of DNA. Just a reminder, what direction is DNA built in? 5' to 3', 3'. That's a key piece of understanding what happens in a replication bubble. Remember this, folks. DNA replicates from 5' to 3'. And when we say 5' to 3', we're talking about new DNA. The new DNA is built from 5' to 3'. Let's watch this in what we've already seen. Notice primase lays down the primer from 5' to 3', and DNA polymerase lays down DNA from 5' to 3'. Even DNA polymerase I removes the primers from 5' to 3' and replaces them with DNA from 5' to 3'. All right, we're going to start at the very beginning. And the very beginning of DNA replication is the origin of replication. This is a location on the DNA where DNA replication starts. And DNA replication progresses in both directions away from the origin of replication. So watch helicase come into the origin of replication and form a replication bubble that gets bigger in both directions away from the origin of replication. And as this is happening, new DNA is built in both directions following DNA helicase away from the origin of replication. New DNA is built in each of the 4 quadrants of the replication bubble. And this happens slightly differently. In 2 of the quadrants, the DNA is built in what's called leading strands, and the other 2 quadrants, the DNA is built in what is called lagging strands. Let's first look at the leading strands. Leading strands are built as one continuous piece of DNA, and this has to do with the fact that DNA is built from 5' to 3', and it is built away from the origin of replication. Let's watch the leading strands being built. So notice, closest to the origin of replication, primase lays down the primer from 5' to 3', and then DNA polymerase builds DNA from 5' to 3'. In this other quadrant, the same thing happens. The new DNA and RNA is built from 5' to 3'. These areas are called leading strands because of the direction they're situated. They can be created in one continuous piece. Things happen very differently in the other 2 quadrants. The other 2 quadrants are built as what are called lagging strands. And because of the situation and their directionality, they have to be built in small pieces. Let's watch the lagging strands being built. The lagging strands are still built from 5' to 3', and they're built first closest to the origin of replication. But notice they have to be built in these small little fragments to enable 5' to 3' building and to enable the fragments to be built closest to the origin of replication, and then to continue to be built further away from the origin of replication as that replication bubble grows in both directions away from the replication bubble. Let's watch that one more time. Notice it's laying down from 5' to 3', closest to the origin of replication. And then a new snippet is built from 5' to 3', a little further away from the origin of replication. Let's look at the other quadrant. Again, being built from 5' to 3', closest to the origin of replication. And now the next fragment will be built a little further away, still built from 5' to 3'. Now those little fragments on the lagging strand, we named them because don't we love to name everything in biology? And we name them Okazaki Fragments. So looking at the lagging strands, each of those little fragments we call Okazaki Fragments. Then the primers are removed and nicks are repaired. Let's watch DNA polymerase 1 remove the primers, removing the RNA, replacing it with DNA. And then finally, DNA ligase repairs the nicks so that we get continuous brand new strands of DNA. Thank you for joining me. Remember to please subscribe to the channel. And don't forget, if you're studying this topic, there are free flashcards in a link in the video description.